Okay, let's dive right into this explainer. If you've ever stopped to wonder how your body, or literally any living thing on Earth, actually functions at the most basic level, you're in the exact right place. We're about to take a microscopic factory tour to uncover the incredibly complex, hidden universe that is operating inside of you right this very second.
Here is our roadmap for today. First, we'll look at the discovery of the cell. Second, the cell's security system. Third, we'll go inside the control center. Fourth, we'll meet the organelle workforce. And finally, we'll explore how cells multiply. All right, Section 1, Discovery of the Cell. Let's unlock this microscopic world. So picture this. It's 1665. A scientist named Robert Hooke places a super thin slice of tree bark, just regular cork, under a primitive microscope he designed himself. Looking through the lens, he notices it looks almost exactly like a honeycomb. Fascinating, right? Fast forward a bit to 1674, and Leeuwenhoek actually discovers free living cells swimming around in pond water. By 1831, Robert Brown locates the nucleus, and by the late 1830s, Schleiden and Schwann present the famous cell theory. This was a massive game changer, suggesting that absolutely all plants and animals are built from these exact same foundational units.
The crazy part is, when Hooke saw those little honeycomb compartments, he named them cells, which actually comes from the Latin word for a little room. It's such a cool historical footprint because that was the very first time anyone realized living things are made up of separate, tiny building blocks. And hey, it's a term we still use every single day.
Moving right along to Section 2, the cell's security system. We're talking plasma membranes and cell walls. To really understand how these microscopic security gates work, we need to ask ourselves a pretty practical question. How do water or materials actually get past the plasma membrane? Imagine just dropping a living cell into a highly concentrated saltwater solution. What do you think happens to it?
Well, the answer beautifully illustrates the process of osmosis, which is just the diffusion of water across that selectively permeable membrane. In a dilute hypotonic solution, water rushes right into the cell, making it swell up like a water balloon. In an isotonic solution, the flow is perfectly balanced. But in a highly concentrated hypertonic solution, like that salt water we just talked about, more water leaves the cell than enters it. And the result? The cell drastically shrinks.
But nature is incredibly adaptable. When you compare plant cells to animal cells, you notice plants have an extra layer of security. They have this rigid outer cell wall made mostly of cellulose. So if a plant cell takes in way too much water, this wall just pushes right back with equal pressure, which totally keeps it from bursting. Animal cells, on the other hand, they completely lack this wall, making them way more vulnerable. They just pop under that kind of extreme swelling.
Let's step into Section 3, inside the control center, the nucleus and DNA.
Think of the nucleus as a highly guarded vault. It's surrounded by a double-layered membrane featuring these tiny little pores that tightly control what goes in and out. But inside is where the real magic happens. The nucleus houses your chromosomes, which are made of DNA and protein. And your DNA? That is the ultimate master blueprint. It contains functional segments, genes, that dictate absolutely everything the cell is going to become, directing its chemical activities and its entire reproductive cycle. Now, here's a fun fact. Not all cellular factories actually have this distinct walled-off control room. Primitive organisms, like bacteria, which scientists call prokaryotes, they basically have an open floor plan. Their genetic material just sort of floats around in a poorly defined region called a nucleoid. Eukaryotes, though, which includes plants, animals, and, well, us, are larger, way more complex, and feature that beautifully defined membrane-bound nucleus. On to Section 4, the organelle workforce. This is all about the division of labor. First up on our factory floor, we have the incredibly efficient manufacturing and shipping departments of the cell. I'm talking about the Endoplasmic Reticulum, or ER for short, and the Golgi apparatus. So the ER is this massive network of tubes and sheets. The rough ER actually looks rough under a microscope because it's completely studded with tiny little machines called ribosomes, and their whole job is to manufacture proteins. The smooth ER doesn't have those bumps. Instead, it makes fats and lipids. And crucially, in your liver cells, the smooth ER is what works completely overtime to detoxify poisons or even process that headache medication you took this morning. Once all that stuff is manufactured, the Golgi apparatus modifies it, boxes it up, and ships it out. Next on our tour is the specialized sanitation crew that keeps the cell's environment perfectly clean, the lysosomes. Lysosomes are basically these little sacs filled with incredibly powerful digestive enzymes capable of breaking down organic material. They are super effective at digesting foreign bacteria or worn-out organelles. But they carry a pretty dark, dramatic nickname: suicide bags, because if a cell is fatally damaged, the lysosomes will literally burst, releasing all their enzymes to completely digest and destroy their own cell. It's basically the ultimate sacrifice to protect the rest of your body from a rogue or dying cell. Of course, none of this manufacturing, shipping, or cleaning machinery works without a massive amount of energy. And that brings us to our power generators, the mitochondria. You've probably heard the phrase, but mitochondria are undeniably the powerhouses of the cell. They have this deeply folded inner membrane that hugely increases surface area, so they can generate energy in the form of ATP, which is basically the universal energy currency of life. But here is a truly fascinating anomaly. Unlike the other organelles, mitochondria actually possess their very own independent DNA and ribosomes. So they are basically running their own mini-factory inside the larger cell to make their own proteins. How cool is that?
Before we leave the factory floor, we definitely need to acknowledge a few departments that are exclusive, or at least highly specialized to our plant cells: plastids and vacuoles. See, plant cells don't eat food, they literally have to make it. That's where plastids come in. Chloroplasts are a type of plastid packed with green chlorophyll to perform photosynthesis. Another type, leucoplasts, acts as a sort of warehouse for starches and oils. Furthermore, plant cells require a massive central storage sac, or vacuole, which can actually take up a staggering 50 to 90% of the entire cell's volume. Think about that, a storage tank taking up almost the whole room. It stores cell sap and gives the plant cell the crucial rigidity it needs to stand up tall toward the sun.
Finally, Section 5, how cells multiply. Let's look at growth and reproduction.
So there are two entirely different blueprints for cellular expansion. First is mitosis. When you heal from a paper cut or, when you were growing taller as a kid, that's mitosis. One mother cell dividing into two completely identical daughter cells, maintaining the exact same number of chromosomes. But for biological reproduction, the whole blueprint changes to meiosis. This is used exclusively to form reproductive gametes, like sperm and egg cells.
In meiosis, the cell undergoes not one, but two consecutive divisions. And because of this double division, one mother cell ultimately produces 4 entirely brand new daughter cells. But here is the brilliant, critical part. These new cells only have exactly half the number of chromosomes of the mother cell. It perfectly halves the genetic material, preparing it to combine with another organism's gamete to create a completely unique new life form.
It really is an incredible, elegantly designed system. Back in 1855, the biologist Virchow expanded cell theory by stating that all cells must arise from pre-existing cells. But understanding all this mind-boggling microscopic complexity leaves us with one profound, lingering mystery. If every single living cell today, with its security gates, its complex DNA command center, its busy organelle workforce, was meticulously copied and divided from a pre-existing cell, where exactly did the very first cell come from? I'll leave you to ponder that one. Thanks for joining me on this explainer, and keep that curiosity alive.